Okay, so what we're going to do is look at increasing rates of reactions, carry on looking at that today. So yesterday we looked at what would happen if we increase the surface area of a solid and temperature. And then we're going to look at now what would happen if we changed any of the others as well. So quick recap from yesterday, we said that basically if we increase our surface area of our solid, we increase our chance of collisions. If we've got more collisions happening, that's happening because we've got more particles exposed, therefore a rate of reaction increases, because we can increase our rate of reaction by increasing our chances of collisions by putting more particles in or putting more energy into the system. So that's how catalysts work. We increase their surface area, so we have them as powders because they have more of their particles are exposed than if they were one big solid. Other one that we looked at was increasing temperature, and the idea is if we increase the temperature, it increases the rate of reaction because particles get more energy, so they can overcome that activation energy that we talked about before. And we looked at the Maxwell-Boltzmann distribution of molecular energy, and that's basically where it says not all molecules have the same amount of energy and velocity. Most of them have this intermediate velocity, and some have very low and some have very high. Now, if we add in, let's get rid of them for a minute, activation energy is over this end. So actually, we don't actually have that many of these molecules with sufficient energy to overcome that energy barrier. So that is an effect that will affect our rate of reaction, and the amount of molecules we have that can that have at least the activation energy changes our rate of reaction. So therefore, if we then increase the temperature, what we then have is this area under a graph here. We've got an increased amount of particles here, increased amount of molecules with the activation energy. So molecules with sufficient energy to overcome the energy barrier, that amount has increased. So increasing the temperature gives particles more energy, and that energy is greater than the activation energy. So more reactants are able to overcome the energy barrier and form products, and therefore a small rise in temperature can lead to a large increase in rate. So that is what increasing temperature would do. The third way we can increase a rate of reaction does similarly sort of link with that first one we talked about, which was surface area, and that's adding a catalyst. Catalysts work in a little bit of a different way. You might remember a bit of how they work from GCSE, especially when you've done about enzymes as a biological catalyst. And so catalysts at GCSE, we regularly just say are a lower activation energies, or they speed up rates of reactions. And so what we're going to do is we're going to actually talk about how they work. So what catalysts do is they actually provide an alternative reaction pathway that has a lower activation energy. So obviously, when we think about the concepts of energy, and well, we have a set amount of activation energy, we need to hit it. It's a bit strange. We go, oh, a catalyst lowers it. So actually, what the catalyst does is it actually provides an alternative pathway. And this alternative pathway still has an activation energy, but it has a lower activation energy than the original pathway. So that is how we then start to increase that rate of reaction. So if you think back to the last graph here, if we have changed it so we've got a lower activation energy, our activation energy then might be coming in over here. So we've got an even larger amount of molecules that are able to hit that activation energy barrier. So it's working with this Maxwell-Boltzmann distribution of molecular energy with the catalyst. So if we decrease the activation energy, the idea is that more particles will have that sufficient energy to overcome the energy barrier and react. Catalysts also remain chemically unchanged at the end of a reaction, so catalysts can get reused again and again and again. So here's a perfect example of a reaction without a catalyst and then with a catalyst. So we using one of them enthalpy diagrams again, and we've got our enthalpy of our reactants, enthalpy of our products. This is our activation energy up here, and this is our delta H enthalpy change down here. And this is a very, very high activation energy. And you can see that we need a lot of energy. And if we were to basically go and put that Maxwell-Boltzmann distribution of molecular energies over the top, we would be, well, at this point, we'd be at very, very small number of molecules with sufficient energy to overcome the energy barrier. It's going to be slow. It's not going to be very great. If we can increase it and change it, well, if we look at this catalyst, this is what we can do instead. So we get this alternative reaction pathway. Sometimes they have more than one activation energy involved. So this one actually has 2 activation energies involved. So instead of going all the way up here, particles that we actually have 2. So we have to reach this activation energy. And then once we've reached that one, the energy goes down, and then we need to have another activation energy here. So activation energy has decreased a huge amount. So this whole area here, the particles in this region originally would not have been able to react. So we'd have had such a small amount of particles able to react. Now, all these particles in this region can now react. So we've increased the amount of particles that can actually react and overcome that activation energy. So again, enthalpy change isn't going to change. Enthalpy changes aren't affected by this because our enthalpy of our reactants and our enthalpy of our products aren't going to change. So our delta H, our enthalpy change, isn't going to change. It's just our activation energy. So our overall enthalpies aren't going to change. It's just activation energy, and that's because we've got an alternative reaction pathway. So we haven't lowered the activation energy of that pathway. The catalyst has added something in. It's given us an alternative route for this reaction to take place, which lowers the activation energy, and catalysts remain chemically unchanged at the end. So it's not changing a chemical reaction, it's just giving all those chemicals an alternative route to get to where they were going anyway, and we can keep reusing them again and again. Same as enzymes being a biological catalyst, enzymes can be used again and again and again. So they're really, really useful in this way. So here we go, adding a catalyst. So the area under the curve beyond EA corresponds, again, like I was saying, the number of molecules with sufficient energy to overcome the energy barrier and react. So if a catalyst is added, our activation energy is lowered. So on this graph, our activation energy will move to the left. So now we've got, because it's moved to the left, we've now got some more molecules with sufficient energy to overcome the energy barrier. I mean, we're not all the way like over here, but that doesn't matter. We've still got a good chunk more than we had before. So we've got all of these molecules now. So it's not just these, it's all of these. So by lowering the activation energy, this has resulted in a greater area under the curve after EA, after activation energy, which shows us that there's more molecules have energies in excess of the activation energy, so they can overcome that energy barrier. So we're going to have a little quick review of catalysts. So catalysts work in a way by providing an alternative reaction pathway of lower activation energy. That's a pretty key definition really that we need to know. At A level, and it's that word alternative that's really, really key. So it's not just that it provides a lower activation energy, it provides an alternative reaction pathway with a lower activation energy. That's really, really important. And I know I keep repeating it. And that's that sort of key difference of terminology between GCSE and A level. And your answer at GCSE wouldn't be sufficient to get a mark at A level anymore. So using catalysts are great because they avoid the need to supply extra heat. So they're safer, they're cheaper. Obviously, if we're increasing the heat of something, we need to be supplying that heat to it. So the more heat we need to be adding to a system, the more money it costs. Again, safer as well. Obviously, we know lots of chemicals are flammable. Heating them up to a certain point isn't always useful. Also, if we heat them up too far, we can start to change things and not necessarily have the chemical reaction we want. So it can get a bit, we have to be very careful with temperatures and be very much on the exact point. Whereas in catalysts, we don't have to have that worry and that control. We can just stick the catalysts in and it goes. Catalysts remain chemically unchanged at the end of a reaction. So here's some types. We can get homogeneous catalysts, and these have the same phase as reactants. so CFCs and ozones. We can also get heterogeneous catalysts, and these have a different phase to our reactants. For example, iron in the Haber process. We haven't done the Haber process yet, but when we do, we'll look at that again and we'll review catalysts again. This is really, really important. Okay, so catalysts do not affect the position of any equilibrium. We haven't done a whole lot on equilibria at the moment yet, but if you think back to GCSE, equilibria is obviously the point of where we have that balancing between them, we have equal amounts of both products, reactants, and obviously catalysts don't affect that. They can affect the rate at which equilibrium is attained, so they can speed up the rate at how quickly equilibrium is attained, but it doesn't affect the position of equilibrium. So when equilibrium will occur, where there's that balance, it will always happen. It just might happen quicker or sooner than it was before because we've added a catalyst, so it happens quicker, but it doesn't change the point of equilibrium. So a lot of money and time is spent on research into more effective catalysts. The savings can be really, really huge and really dramatic that you can make just by adding a catalyst. We can be saving thousands, millions, depending on what it is. Places that have catalysts in, things like washing powder, biological washing powder has, they have enzymes in basically, and they have biological enzymes in there, and they're used to help get rid of all the dirt and muck and grime and sweat on your clothes, which is great because actually we can get rid of all that stuff on like a 90, 100 degree wash, but one, that's really hot. Two, that costs a lot of money to heat our washing machine up to that temperature. Three, our clothes would have to be in the washing machine for a long time. Four, our clothes would shrink. Washing clothes at a really, really high temperature isn't the best for them. The lower the temperature you can wash them at, the better they stay, and the better for the environment it is because it doesn't require as much energy, because we're not heating water up as much. But obviously we had that sort of handoff at one point where it's like, well, this is better for the environment, better for my clothes, but my clothes still smell and they still look mucky. Add in a biological catalyst, and actually we've got that balance. By adding in these enzymes, we've been able to lower the temperature. We've been able to lower the time they spend in the washing machine because we have those catalysts in there breaking everything down for us quicker. Whilst we say the catalysts remain chemically unchanged at the end of the reaction and we can use them again, they do need to be changed regularly as they can get poisoned by other chemicals, so they can get changed slightly. So we say they remain chemically unchanged. Basically, they can get, if it's an enzyme, it can become denatured by other chemicals that are present, and other catalysts can get poisoned, so they just don't work effectively anymore. So we do have to keep adding them into a system. And catalysts are used in that finely divided state to increase the surface area. Like we said right back at the beginning, large surface area increase the rate of reaction. The smaller your catalysts are, the more faster they're going to work because we've got more surface area, we've got more collisions happening. More chemicals are going to be in contact with those catalysts. So catalysts, why do we use them? Probably lots of things I've already said. We use them in industry because they allow reactions to take place at lower temperatures, and that's the massive thing. We're not having to have these high temperatures. Saves energy. It saves loads of energy because we have a lower activation energy, so we don't need things to be as hot. Because we don't need things to be as hot, we're not heating them up as more, we're reducing our CO2 output, which is great for the environment. They also enable different reactions to be used. So we often get better at atom economy, which we talked about weeks, probably months ago, really, atom economy, we talked about, and yield and how much we get from the reactants, how much do we get from the products, how much is actually still there. We get better at atom economy, actually, using catalysts. We don't get as much waste using catalysts. So it helps there a lot as well. And they're often enzymes, like I was saying. They're often enzymes because enzymes generate a specific product and they operate effectively at room temperatures, which is great. Enzymes love room temperature. And they have great economic importance in industrial production of these things, so such as polyethylene, sulfuric acid, ammonia, and ethanol. So they're used a lot for making those. And they can also reduce pollution. So catalytic converters in your car, they actually reduce the pollution of the fumes coming out of your exhausts. So all cars have them in now, but there would have been, I say all cars, most vehicles will have them in of a certain age. And certain areas, if you go to some big cities, especially in London, they have like low CO2 emission zones. So if you have a car that doesn't have a catalytic converter inside it, you will get charged. I don't think it's technically a fine, but you get like a charge given to you. It's basically a fine because you're driving in a CO2 area, and you're allowed to, but you're basically told if you're going to drive here and you don't have low emissions, we're going to charge you some money because you're adding to our emissions. So it's areas that are already highly polluted in a city area. So there's lots of low CO2 emission areas in London. You can also get some, I think there's some in Manchester and Birmingham as well. There's probably some all over the country, but they're places I've seen them in. So that's our catalyst overview. Another thing we can do to increase the rate of reaction is we can shine lights. This doesn't work for everything, unfortunately. So we can't just go around shining lights on everything, hoping that rates of reactions are just going to magically increase. It's only for certain reactions. And it needs to be a certain type of light source as well. So this is where we start to get quite picky with it. So shining a suitable light source onto some reactants increases the rate of reaction. So the light, which is often UV, provides energy to break bonds and initiate a reaction. So the greater the intensity of the light, the greater the effect. So an example, reaction between methane and chlorine. And so that's an alkane. Darkening of silver salts, which we use in photography, and then the reaction between hydrogen and chlorine. So we're going to look at the hydrogen and chlorine one. So hydrogen, its bond enthalpies, the HH bond is 436, chlorine to chlorine is 242. So we have initiation, propagation, and then termination. So basically what we do is by adding that UV light, we increase that rate of reaction because it provides some energy to break those bonds and initiate the reaction. Otherwise, they've got quite high bond energies that would actually need to be broken. So we're adding that light to it that actually helps break those bonds and helps that reaction take place. The other thing we can do to increase the rate of reaction is increase pressure. Pressure, obviously, we're talking about gas particles in this situation. And so if we increase the pressure this basically just forces all of our gas particles closer together. The closer together they are, the more likelihood there is that a collision is going to take place. So frequency of collisions is going to increase. Therefore, our rate of reaction is going to increase as well. Many industrial processes occur at high pressure to increase the rate, but unlike catalysts, increasing the pressure can affect the position of equilibrium and it can affect yield. Catalysts don't do that, increasing the pressure does. So the more particles that we have in a given volume, the greater the pressure we have. The greater the pressure, the more frequent the collisions. The more frequent the collisions, the greater the chance of reaction. It's your basic understanding of pressure, but the biggest issue there is equilibrium and yield. We could increase the pressure to really high values, but our equilibrium could shift so much and our yield could be so poor that it's not worthwhile. So we have to be really careful when we're dealing with pressure that actually we're finding a sensible place to put it. Also, we have the issue with pressure. If we increase it too much, it can actually be very dangerous as well, sort of like the issues that we have with temperature. And so we have to be quite careful with pressure as well. Another thing we could do is increase the concentration. If we increase our concentration, we're going to get more collisions. If we've got more collisions happening, we've got an increased rate of reaction. So Image on the left, we've got a low concentration of particles, we've got fewer collisions. Image on the right, same shaped area, but we've got a high concentration of particles, which means we have more collisions. However, this is a big however, it seems logical, well, I'll just stick more in, so then I can have faster rate of reaction. However, increasing the concentration of some reactants can have a greater effect when increasing others. So we have to pick which ones we're going to be increasing and which ones we aren't going to be increasing. I mean, if we just increased everything, it has very minimal effect. So we have to be quite careful deciding actually what are we going to increase, what are we going to decrease. So rate change during a reaction. So reactions, if they hit the activation energy, if our particles can breach that energy barrier and a reaction is triggered, they are the fastest at the beginning and then they slow as the reactant concentration drops. It makes sense. And we've got all these reactants and then they're going to get used up. So as they're used up, the reaction will start to slow because we have less collisions. The pressure will decrease. Our concentration will decrease. So when we're starting to have that, so obviously the reaction rate slows down. So here we go. In a reaction such as A plus 2B gives us C, the concentrations might change as shown. So A and B are our reactants, C is our product. So reactants A and B, their concentration decreases with time. Product C, concentration increases with time. It makes sense. The steeper the curve we have, the faster the rate of a reaction. So as we can see, B's got a steeper curve than A. Reactions always start off quickly because of a greater likelihood of collisions, and reactions slow down with time as there are fewer reactants to collide with each other. And you can see they're all starting to slow down at the same points where they're crossing on the graph. So A and B are our reactants, C is our product. So we can measure the rate of reactions. And so this is an experimental investigation that we're going to look at. And there's lots and lots of different ways. Main one that you probably did at GCSE was a disappearing cross one. So it's sodium thiosulfate into sulfuric acid in a conical flask, and you put that X on the table, and you swirl your beaker around until you can't see, conical flask around until you can't see the X anymore, and you stop because that's the reaction completely finished. And you can do quite similar things with titrations as well. Massive issue with them is they're not the most reliable. We're using human error and things, but that's sort of, and then you would repeat it multiple times, changing the temperature or changing the pH or changing concentration. But it all depends really on the type of reaction that we're doing and the properties of the reactants of a product. So an example could be extracting a sample from a reaction mixture and analysing it by titration. This is often used if an acid is one of the reactants or products, so there's quite a regularly done one. We can use it doing titrations. I think this one was used at GCSE as well. And you're basically adding an acid to an alkali, and you're waiting until it hits a point that the alkali becomes acidic. So we're adding enough of one product to the other so that then it overpowers it. So we're looking where that point of equilibrium is. You can use a colorimeter or UV visible spectrophotometer to see rates of reactions using different colours. Quite one that's used quite regularly in biology is when I spoke to you about the membrane, alcohol concentration or temperature affecting the permeability of the cell surface membrane. So basically what we're looking at there is what bonds have been broken as we increase the temperature. And what you do is you do this to some beetroot, and the idea is the more beetroot stain that gets through and into the liquid, the more the cell surface membrane has been broken down, because the pigments can get through. So therefore, the ones that have been broken down the most would have a darker colour, and ones that have been broken down the least would have a light colour. You can try and observe this with your eye, but it's not the most practical or sensible way of doing it, or scientific. So we can use a colorimeter, which basically records the level of light going through just some clear distilled water, and then you then put in all your other different samples, and then it gives a reading based off that of how much light can penetrate through there. UV spectrophotometer works in a similar way. If we're dealing with gases, we can measure the volume of gas that's been evolved. If we're dealing with reactants or products that are conductors, we can actually measure the change in conductivity. There's loads and loads of other ones that you can find in textbooks and revision guides, and it's sort of quite endless, really. Lots of these different ways we can investigate these. So then we can measure the rate. So if we have a look at this graph, we can look at how much concentration changes with time, and that is equivalent of velocity. So basically, we've got this graph, we've got concentration against time, we've got our gradient. And what happens is the slope of the gradient of a curve gets less as the reaction slows down, and we know that. But we can work out the different gradients of the graph using y divided by x, y, and x on our drawing the triangles on. So what we can do is the rate of change of concentration is found from the slope, the gradient of the curve. The slope at the start of reaction will give the initial rate, and then as the slope gets less, showing the rate is slowing down as the reaction proceeds. So actually we'd have our initial rate of reaction, and then we'd have a final rate of reaction, so we can show that it's changing as we're going. So let me just pause this. But yeah, so we're going to look at lungs and gas exchange. We're going to look at mammalian lungs today, and then tomorrow, before we look at I can't remember what else we're looking at tomorrow. It could be the immune system. We're going to look at insect respiratory systems, fish, insect fish. I think that's it, insect fish. It could be something else as well. Insect fish, maybe a frog. So we need to know 3 others as well. I'm sure it's insect and fish. Maybe it's insect, fish, and mammal. I'm not sure. I'll double check tomorrow. But at the minute, we need to recall the structure of a mammalian lung, know the relevance of surface area, concentration gradient, and Fick's law in relation to gas exchange. And then we'll look at sticky mucus increasing the chance of lung infections and reducing gas exchange. It's actually quite nice to do this one actually after that bit of chemistry, because we actually link in with each other quite well, because we're sort of talking about rates of reactions, rate of diffusion. So whilst diffusion isn't necessarily a reaction, it's affected by very similar things to a rate of reaction. So a bit of an overview where we are. So genes play a major role in our health. So in the world, we have over 10,000 genetic disorders which can be diagnosed, from minor disorders such as colour blindness to much more serious disorders. So we can have things such as cystic fibrosis. We're going to have a little talk about cystic fibrosis and how it affects the lungs during this last half hour about this. But cystic fibrosis is a genetic disorder. It's caused by mutation on an allele. It's inherited, it is recessive. So when we go more into genetics, if you think back to GCSE, dominant and recessive genes and alleles, dominant genes, you only need to have one of the alleles present to have that. So if you think about eye colour, actually, if we do a little quick recap actually of this, we think about eye colour, brown eyes we will have as B. The allele for brown eyes is B, and that's going to be brown. The allele for blue is going to be little b. Blue, and that is because brown is dominant and blue is recessive. So If, for example, both your parents had brown eyes, one parent might have the gametes BB, and the other parent might have this. You would never, ever basically end up with blue eyes, and I'll show you why. You could have from one parent and the other BB. So you would have your phenotype would be brown, whilst you might have the alleles for both for brown. You could have BB again. You could have BB again, and then finally you could have BB. This is the only one that carries the blue allele, but blue being recessive means it wouldn't be shown, so it would have to your eye colour, your phenotype would still be brown. But if we clear that up, let's have a look at if the parents had this. I'll do it as a Punnett square. Okay, if you ended up with this, your phenotype would be brown eyes, and you're carrying the alleles, both of brown eyes dominant. If you ended up with this genotype, your phenotype would be brown eyes, because brown is dominant, but you'd carry the recessive allele for blue eyes. Exactly the same if you had this genotype, because they're the same, and this genotype would cause you to have the phenotype of blue eyes, because for a recessive gene to be shown in your phenotype, you have to carry both copies of a recessive gene, and that's how cystic fibrosis works. You need to have both copies of a recessive gene. So we have cystic fibrosis. And cystic fibrosis basically affects our mucus in our systems. So it's why we'll talk about it with lungs. The mucus in our body just really great jobs. It's great for our immune system. It keeps pathogens out of our bodies. We also have mucus in our digestive system to help enzymes, but obviously they can be an issue if we have too much. We also have mucus in the reproductive systems, but again, too much can cause an issue. So cystic fibrosis is when we have not just too much, but it becomes more viscous and more sticky than normal. So there can be lots of issues with that. Oops. Why is that making Huntington's disease is also another genetic disorder. Sickle cell anemia. Sickle cell anemia is where red blood cells are shaped like a sickle. If I'm not sure what sickle is, it's what people used to use probably medieval times. It could have been sooner, I'm not sure. To cut wheat, and it looks a bit like that, and it would have a handle that you'd hold, and the sickle part is just this part. It's almost like a bit like a crescent moon. And so basically in sickle cell anemia, your red blood cells are sickle shaped. The issue with that is obviously they're smaller, they can't carry as much oxygen, so it's harder for your body to get enough oxygen that it needs. Haemophilia as well, which is a blood clotting disorder. So since these disorders can be passed from parents to offspring through genetics, we say these are inherited. So cystic fibrosis, and this is the most common genetic disorder amongst white people. 1 in 2,400 babies are affected. Now, genetic disorders, you will have some that are more common between different races of people than others. It just depends on where they sort of originated from. And so it can be more prevalent in certain races of people. It doesn't necessarily mean that people of a different race can't have that, but it's just would be classed as most common. So again, so amongst white people, cystic fibrosis is one of the most common. So 1 in 2,400 babies are born with cystic fibrosis. The recessive allele is actually carried by 1 in 20 white people. It can be carried by people of other races, but it wouldn't be as in such high prevalence, and the ratio would be a lot smaller. So pre-1940, 80% of sufferers died before the age of 5. But now life expectancy is around 35, but it can even be older, 40 to 50. And that's just with the advances in medicine and us being able to treat people with cystic fibrosis a lot better. We didn't identify the gene that caused cystic fibrosis until 1989. And the normal allele codes for a protein that is important in the cells lining the airways, lungs, reproductive systems, and gut. And the faulty allele basically results in a thick dehydrated mucus being produced in these areas. The issue with the thick dehydrated mucus is is great. Like in our respiratory system, we have loads of mucus to trap pathogens. It's supposed to trap them, white blood cells can come in, phagocytosis can happen, and then we can get rid of them out of our body. If it's too thick and it's dehydrated, phagocytosis and actually just getting rid of that mucus is a lot harder to happen. So actually it becomes actually a perfect place for bacteria and pathogens to grow. It also in our lungs, and if it's really, really thick, oxygen can't get through it. And obviously we need oxygen and carbon dioxide to diffuse through that. The mucus will actually clog areas up, so it reduces our surface area into our lungs a lot more. So because cystic fibrosis is inherited, it runs in families. So an individual with a family history of a disease may well be carrying this cystic fibrosis allele. So what we'll actually look at some points is individuals, if they know that someone in their family might have cystic fibrosis, they might look at getting a genetic testing first to see if they have carry the allele for if they then want to have a child in case they could pass it on. So we're going to look at later on when we do a bit more genetics how it might affect the child. So the lungs, let's have a look at the lungs. What is happening with that picture? I don't know. Right, the gas exchange system. Because one of the effects of cystic fibrosis on mucus production, we're going to look at the gross and microstructure of the human lungs. The gross structure is basically back at that point where you would be at GCSE. So we've got our trachea coming down. Let me put my pointer on actually. Where's that? Use a pointer. So we've got our trachea, which obviously joins up with the nose and the mouth, and our trachea comes down and breaks into two halves because obviously we have two lobes of the lungs, which are our bronchus. So we have our trachea and our bronchi. Individual bronchi is a bronchus, so that's the plural, and sorry, and that's the difference between the plural and the singular. Bronchi is plural, bronchus is singular. So we switch into bronchi, bronchus. Then we have our bronchioles, which are our next strand off. Then we go down to the alveoli. So this is our right lobe. This is our left lobe, same as the heart. They're the opposite way around on an image. So if we have that image up to our chest, the right side would be on the right, left would be on the left. So basically, any gases that are coming through or out, so carbon dioxide and oxygen, for example, travel through the trachea, through the bronchi, now the bronchioles, into the alveoli. Obviously, we have something called pleural fluid at the bottom, so we actually have a lot of fluid involved bottom of our lungs to help our lungs expand and retract. If people can have punctured lungs, so we can say the lungs are punctured or deflated, the pleural fluid's often involved. If people say we've got fluid on the lungs, it can either be one of two ways. It can be that we've got fluid in some other way, but often there can be issues with the pleural fluid, and there might be like an injury to the lung where some of that pleural fluid has then got in. And then obviously the diaphragm sits underneath, and the diaphragm plays a huge part in helping our lungs to contract and expand. And then we have the alveoli at the end. They look like little grapes. So this is sort of the microstructure now. So that was our macrostructure. This is the whole lung. This is then when we look into it in a bit more detail. So obviously, the whole point of the lungs is to get oxygen into the body, into the blood, and carbon dioxide out of the blood, out of the body. And so we often don't draw it like this when we're looking at the macrostructure. But when we look at the microstructure, we have a bronchiole, and next to the bronchiole, we have a branch of the pulmonary artery, and they run right next to each other. Pulmonary artery, that is the artery that is going away from the heart. It's going to the lungs. So that's going to have deoxygenated blood in it. It's the only artery that carries deoxygenated blood, and that's because it's come to the come to the lungs to come and get oxygenated. We also have a branch of a pulmonary vein at the bottom, so once it's oxygenated, it can then go back to the heart to then get pumped back around the body, and all that oxygenated blood can go around our body. So they run next to each other, and they sit next to each other because actually we need them next to each other so we can then have this diffusion happening. So we have the bronchiole and the branch of pulmonary artery next to each other. And then as the pulmonary artery gets thinner and breaks into different branches, they go into arterioles, and then it basically goes into the capillaries and the capillary beds. And basically the capillary beds sit all over like a big mesh net even around the outside of our alveoli. And that is where that gas exchange will be happening. Our carbon dioxide will be leaving, our oxygen will be entering, and that's how we're having that switch over there. And our alveoli, if we cut them open, look into them, they are air gaps. They are empty air sacs. They've got connective tissue around them, but they are empty. They are air, so they can fill as much oxygen as possible. So, I don't want to show it 5 times. Alveoli. What is really great about alveoli? Well, alveoli are fantastic for exchange surfaces because it has an enormous surface area. So they're made up of 70 cubic meters, which is just fantastic, really, because actually, and that's why we have all those rises and those ridges. If we sort of go back to this picture, we have all those lumps and bumps around them. We've got these nets of capillary beds over the top and around them and underneath. And so we just increase the surface area. Like we always talk about, increase the surface area, increases the rate of reaction, increases the rate of diffusion, it increases the rate of osmosis, it increases the rate of active transport. Larger surface area increases rates of reaction, increases gas exchange as well. Increases gas exchange because we have a larger surface area for those gases to actually exchange on. There's more points for them to exchange. They've got a moist lining for dissolving gases. Basically, the way that the lungs work, if they were dry, it wouldn't work. They wouldn't work fully. So those gases need to dissolve, and the way that they work and the way that our body works, we need it to be moist. We need there to be actually that mucus in there as well. That mucus doesn't just trap pathogens; it also helps gas exchange. We need that sort of concentration gradient. They have super thin walls. Now, walls is an interesting word because obviously we're not talking about cell walls, we're just talking about walls of the alveoli. So some people say they have a thin membrane. Sometimes people say they have a thin exchange surface. Any of those is fine, but it's not, it has a thin cell wall because obviously it is a eukaryotic animal cell. They don't have cell walls. So be careful with the use of walls. I know I've put the word wall there. It will be used a lot in exam books, but just be very careful with the use of wall, that you're making it clear that you're talking about the membrane or the surface exchange area being thin, not that it's a thin cell wall. And then the fourth thing that makes it a fantastic exchange surface is that it has got a massive blood supply, which is perfect because that's going to help a concentration gradient for all that diffusion to happen. Here we go. So we've got a really simple image of an alveoli and our blood. So we've got our blood capillary, we've got our blood traveling down, and our carbon dioxide is going to diffuse out. Our oxygen is going to diffuse in. So we've got air out. When we breathe out, lots of it will be carbon dioxide, but some of it will still be oxygen that we're breathing out. Air in, lots of it will be oxygen, Some of it will actually still be carbon dioxide that we're breathing in a little bit of as well, but it's some huge differences. We have a layer of moisture around the outside, well, around the outside of the alveoli, and we've got this blood capillary running along here. And this is where we talk about net flow again. If we go back to diffusion and net flow, it's the net flow of the movement. So gaseous exchange rate and concentration gradient, we need that net flow. Fick's law, we talked about Fick's laws before. So Fick's law, gases are exchanged. Let me get this up. Oh. So lungs, they're fantastic. They've got thin walls. They've got large surface area, which increases diffusion. Large surface area increases the rate of diffusion. Got thin wall, a single cell, reduces diffusion distance. We want the shortest distance possible because then it's going to happen quicker. The bigger the distance, the longer it's going to take, the slower diffusion is going to be. We have, if you reference anything to a surfactant, that it's moist, this stops adhesion, it dissolves gases, otherwise gases can end up adhering to the actual inside lining of the alveoli. And then A mentioned capillaries, blood supply is fantastic because it maintains diffusion concentration gradient. So at the end of each bronchiole are a number of alveoli. They, like we said, increase the surface area available for gas exchange, have very thin walls, and are surrounded by numerous capillaries. Our airways, as you can see, we've got lots of ciliated cells in our airways. Got lumen, got mucus sat there, we've got cilia, we've got our ciliated epithelial cells, we've got goblet cells and a basement membrane. Goblet cells produce mucus, and that's some cartilage there. Oops. Goblet cells produce mucus. So mucus comes out of the goblet cell and is along here, and the mucus gets moved by those ciliated cells. So that's in our airways and things like our trachea. Again, here is our ciliated cells, and we have a gap of the cilia for the goblet cell where it makes mucus for the goblet, for the mucus to actually exit there. Epithelial cells. So an epithelium is a type of tissue that cover the surfaces of animals, organs, cavities, and tubes. They consist of one or more layers of cells that sit on a basement membrane. There are two types that we need to know about. So we have squamous or pavement epithelium, and then columnar or cuboidal epithelium. So this is our squamous, very flat, looks a bit like a pavement. Cuboidal epithelium, columnar, I struggle to say that word, really long, thin epithelial. Sometimes it's quite worth looking at images of lung dissections to sort of help you in a lab at A-level. This would be something that people would do, have a go at lung dissection. What's quite interesting is if you dissect a lung, you take a chunk of it off where the alveoli are, you put it into water, it will float because the alveoli are empty air sacs. So because of that, it has quite a low density, so it's actually able to float. So diffusion. So gases are exchanged from the alveoli to the blood and back by diffusion. So the gas that would move from the blood to the alveoli, and the speed of diffusion is controlled by the surface area available, the concentration gradient, and the thickness of the gas exchange surface. And then quickly at the bottom, this is Fick's law rate of diffusion. So the rate of diffusion is affected by the surface area multiplied by the difference in concentration divided by the thickness of the gas exchange surface. So basically, the thickness of the gas exchange surface, the bigger that is, the more negatively it will impact the rate of diffusion. The bigger the surface area, the bigger the concentration difference, the better. So the human lung's really well adapted to gaseous exchange. It's got large surface area, thin walls, large concentration of gradients. A sticky layer of mucus, the issue with that is it actually can block our airways, so it reduces our surface area. Why is this not working? So therefore, sufferers of cystic fibrosis have a reduced capacity for gaseous exchange, and therefore actually we really struggle to do exercise because they're not having that sufficient gas exchange that most people would have. So what happens is during exercise, someone with cystic fibrosis will become short of breath. Regular exercise is beneficial and can help and improve it, but it would not be at the strenuous level of someone who didn't suffer from cystic fibrosis. I am going to, where are we? There we go. I'm going to stop that there before we go any further. So stop the recording. I will do, why is it not stopping?